Hello friends, welcome to BMH Learning. This lecture is about endomembrane system. So endo means within or inside. That is, endomembrane system is a group of membranes and organelles which are suspended in the cytoplasm of the eukaryotic cells. Now what's its function? The different membranes and organelles of the endomembrane system work together mainly to modify, package, and transport the proteins and lipids. The endomembrane system includes endoplasmic reticulum, vesicles, Golgi apparatus, lysosomes, nuclear envelope, vacuoles, plasma membrane. Although the plasma membrane not technically within the cell, but still it's included in the endomembrane system because you will see it interacts with the other endomembranous organelles. The endomembrane system does not include the membranes of either mitochondria or chloroplast. Now let's discuss the coordinated working and function of different organelles of the endomembrane system with the example of a protein. The very first organelle in our list is the endoplasmic reticulum. The rough part of the ER is studded with the ribosomes. Ribosomes synthesize the proteins. They transfer their newly synthesized proteins into the lumen of the RER, where they undergo structural modifications such as folding or the acquisition of the side chains. Now, for the newly synthesized transport protein to reach its destination, first it will be slipped into the transport vesicle that buds from the membrane of rough endoplasmic reticulum. The protein within the transport vesicle still needs to be sorted, packaged, and tagged so that it ends up in the right place. All these changes take place in Golgi Apparatus, which is a series of flattened membranes. The receiving side of the Golgi Apparatus is called the sys phase. The opposite side or shipping side is called the trans phase. The transport vesicles that formed from the ER travel to the sys phase, fuse with it, and empty their contents into the lumen of the Golgi apparatus. As the protein travel through the Golgi, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins are then tagged with phosphate groups or other small molecules so that they can be routed to their proper destinations. Finally, the modified and tagged proteins are packaged into secretory vesicles that bud from the trans phase of the Golgi. Now, some of these vesicles fuse with other parts of the cell and deposit their contents into them, where they will be used, while other secretory vesicles fuse with the plasma membrane and release their contents outside the cell. This was all. Thanks for watching.